Happy morning, students. As 5 minutes, 5 points, I will try to cover the excretory system. Okay. So, in excretory system, first point is you should know the functional anatomy of kidney. In functional anatomy, you should know there are two nephrons. One is juxtamedullary nephron, other one is the cortical nephron. The cortical nephrons are in a major percentage, around 85%, and juxtamedullary nephrons constitute only 15%. And the cortical nephrons' major role is involved in the infiltration, and they have short loop of Henle. And these juxtamedullary nephrons, which are having long loop of Henle, and they are involved in counter current mechanism. So the counter current mechanism is a mechanism for concentrating the urine. Second point is urine formation. So role of kidney in urine formation. So the urine formation includes three steps. One is glomerular filtration, other one is tubular reabsorption, other one is tubular secretion. So what is glomerular filtration? It is the ultrafiltrate of plasma formed by both the glomerular capillaries of both the kidneys per unit time. The value is 125 ml per minute or 180 liters per day. And what is tubular reabsorption is? Tubular reabsorption is whatever that is filtered, say around 180 liters per day is filtered, but normal urine output is 1.5 liters. So around 178.5 liters. So this much amount of urine which is filtered is reabsorbed. Okay, the reabsorption is very important for retaining very important substances necessary for the body. And coming to glomerular filtration, you have to remember the net filtration pressure. That is a balance between the glomerular capillary hydrostatic pressure, glomerular capillary oncotic pressure, tubular hydrostatic pressure, and tubular oncotic pressure. And this glomerular filtration is directly proportional to the net filtration pressure, and it is equal to Kf into net filtration pressure, where Kf is the filtration coefficient, and this Kf depends upon the capillary permeability and so the capillary permeability in turn depends upon the size of the particle and charge of the particle. And second factor is the surface area of the capillary. So one is the capillary permeability and the surface area of the capillary. So surface area of capillary in turn is decided by the mesangial cells. So these two factors contribute to the Kf value. And in tubular absorption, you have to talk about what are the solutes where they are reabsorbed in various segments. Around 60% of the solutes absorption takes place in the proximal tubule, and in all the segments you have the solute absorption, and water absorption is also taking place in all the solutes. And the third point is the role of kidney in urine concentration. So in urine concentration, the very important structure involved is the loop of Henle, and which is involved in the mechanism called counter current mechanism. So what is this mechanism? This mechanism depends upon the hyperosmolarity of the medullary interstitium. Okay, so when the urine is passing from the descending loop of Henle and going to the ascending limb, there will be active reabsorption of the solutes by active transporters, and this solutes entry to the medullary interstitium increases the osmolarity of the medullary interstitium, and this in turn by osmosis will pull the water from the descending loop of Henle. Again, the urine in the descending loop of Henle gets concentrated, and when this concentrated urine comes to ascending limb, again the solutes will be pumped. So this cycle repeats. So the urine which is leaving the ascending limb of Henle is hypotonic, and this hypotonic urine when passed through distal tubule, again absorption of active absorption of solutes takes place, and when it reaches the collecting duct, it is more hypotonic, and since the medullary interstitium is hyperosmolar, there will be movement of water from the collecting duct into the hyperosmolar medullary interstitium, and this was controlled by the hormone ADH, anti-diuretic hormone. So the final concentration occurring in collecting duct is very important, and it occurs in the presence of the anti-diuretic hormone. So coming to the fourth point, urine acidification. The acidification of urine is very important, that maintains the H+ ion in the extracellular fluid, that it maintains the acid-base balance. So this acidification, very two important structures. One is the proximal tubule, other one is the distal tubule. In proximal tubule, there is a sodium hydrogen exchanger which reabsorbs sodium and secretes H plus ion. It exchanges H plus ion. So H plus ion is secreted into the urine. And in collecting that, you have H plus ATPase pump that will also pump the H plus ion into the urine. So this causes the acidification of the urine. Acidification is very important for removing the non-volatile acids from the body. And we have renal buffers. The first buffer is the bicarbonate buffer, which is filtered, and this bicarbonate buffer will takes the H plus ion and it converts into H2O and CO2, and H2O is eliminated and CO2 is reabsorbed. And this H plus ion in the collecting duct, okay, so that is also buffered by the distal collecting buffered by two renal buffers. One is the ammonium buffer, other one is the phosphate buffer. Coming to the fifth point, that is regulation of BP by the glomerular juxtaglomerular apparatus. The JG apparatus comprises macula densa, juxtaglomerular cells and extraglomerular mesangial cells. Whenever BP falls, there will be fall in the GFR. This is sensed by the sensor called macula densa in the distal tubule, and this sends signals to the JG cells to secrete renin. Renin activates angiotensinogen to angiotensin 1, and angiotensin 1 is converted into angiotensin 2, and this angiotensin 2 is a powerful vasoconstrictor and it increases the blood pressure. So overall 5 points to remember. One is the functional anatomy, other one is the role of kidney in urine formation, urine concentration, urine acidification and BP regulation. Thank you.